Good afternoon friends. Today we will talk about the cell cycle. The cell cycle is that important phenomena by which the unicellular organisms divide into two, and by that they work on asexual mode of reproduction. Even when we talk about sexual reproduction, there are also cells combined together and then they form zygote, which is a diploid structure, and again they will work on mitosis cell division, where the division of cells take place. So, how this cells is equally divided into two, as we can see the mitosis cell division, where you will find the cell having the, suppose any parent cell having X chromosome number, means the cell is haploid and it is divided into two half, where they are making a same cell with X and X chromosome. So, how this is happening take place? The haploid cell will again divide into two haploid cells, as we have read in many books that this phenomena is known as mitosis cell division. So, here in between there is one hidden phase is there, which is called as interphase, by which the genetic material duplicates into 2x, and then that 2x will equally divide into 1x, and 1x means the diploid cell will divide into two haploid cell. So, this is known as mitosis cell division. Now, we will understand that this is only happening because there is a hidden cell cycle taking place. So, what is the cell cycle? We will see here, and we will understand that cell cycle has different phases. The cell cycle has three important phases, that is G1 phase, G2 phase and S phase. So, here we will see one by one. First, we will talk about G1 phase. G1 phase is the longest phase in cell cycle, where the two cells which are forming, this is called as the first growth phase, G1 cell phase in cell cycle. What happened in G1 phase? The RNA and protein synthesize, mitochondria divides and chloroplast also divides. So, these three events take place in G1 cycle, G1 phase of cell cycle, that is RNA and protein synthesis. Second, mitochondria divides. Third, chloroplast divides. Now, we will see that resting phase is called as R phase. After G1 phase, there is a S phase. What is S phase? S phase is also known as synthesis phase, where more DNA synthesized and chromosome duplicates but remain attached. So, S phase is known as synthesis phase, where more DNA synthesized, chromosomes duplicates but remain attached. So, this is all about S phase. The third phase is known as G2 phase. G2 phase, that is also called as second growth phase, where more RNA and protein synthesize, mean to say that more ribosome nucleic acid and protein synthesize, that is called as G2 second growth phase. Now, we will see that after, when we add three of the phases like G1, S and G2 phase, it is known as interphase. As we have talked here, the parent cell is divided into two haploid cells. Parent cell is also haploid, but it is dividing into two daughter haploid cells, because there is a hidden phase which is called as interphase. Interphase is a phase where the genetic material duplication take place. Now, X haploid cell will become 2X diploid cell, which will be equally divided into X haploid and X haploid. So, here we will see that interphase consists of three important phases, that G1 phase, S phase and G2 phase, where we have understand that G1 phase is known as first growth phase, where RNA protein synthesize, mitochondria divide, chloroplast divide, and then there is a S phase, which is a synthesis phase, where the more DNA deoxyribose nucleic acid synthesize, chromosome duplicates but remain attached. And here G2 phase is known as second growth phase, where more RNA and protein synthesize. Now, we will see that in cell cycle one more important phase is there, which is called mitosis, cell division. The cell division take place. So, here we will see that there is a prophase after G2 phase, there is a prophase because genetic material is doubled now. Now, there will be mitosis, cell division. So, we will see that how one cell is dividing to two cell with same chromosome number, like haploid parent cell, both the cells will become haploid. Two daughter cell formation, how it is taking place, because there is a mitosis cell division. The mitosis cell division has four important parts. The first part is known as prophase. The prophase is one of the important part of mitosis cell division, where the genetic material becomes short and the chromatin fibers are there, which will become short heighted and thick. Then the prophase is also divided into prophase, early prophase and late prophase. Then there is a metaphase. Metaphase is that stage where the chromatids or chromosomes are arranged on equator, that is called as metaphase. That is also divided into two parts, like early metaphase and late metaphase. Then we will see the anaphase. In anaphase, the centromere is attached with a spindle fiber and both the loops were separated like this. They used to make a Y fork, chromosome used to make Y fork, and then it will separate, because with the help of a spindle fiber they will migrate separate towards the pole. So, that is the anaphase. Anaphase is also divided into two parts, that is, first is called as early anaphase and late anaphase. Now, we will see the telophase. The telophase is that important phase where the division is now ready. The cell, parent cell will now convert into two daughter cell, because this phase includes karyokinesis as well as cytokinesis. So, we will understand that what do you mean by karyokinesis. Karyo means nucleus and kinesis means division. So, division of nucleus is known as karyokinesis, and that is very important feature of the cell division. In telophase, the karyokinesis means division of nucleus take place into two parts. One single nucleus will divide into two, single nucleus is divided into two nucleus, that is, suppose haploid parent cell, then two haploid daughter cell formation will take place, where the nucleus is equally divided. Then there is a second phase which is called as cytokinesis. Cytokinesis is that process in which the division of cytoplasm take place. The parent cytoplasm is equally divided into two halves, and then the formation of two daughter cell take place with the combination of karyokinesis as well as cytokinesis. So, what we have understand? We have understand that in cell cycle is that very important phenomena where the genetic material of parent cell will duplicate, and by that, with the help of mitosis cell division, it will simply divide into two daughter cell, where it is Xerox copy of the parent cell, very identical to the parent cell, because karyokinesis as well as cytokinesis take place with all the four phases in mitosis cell division. Because after this cell division, the parent cell will singly divide into two daughter cell. Why this is possible? Because there is cell cycle, and in cell cycle there is a interphase. What is interphase? It is a combination of G1, G2 as well as S phase. So, in G1 phase we have understand that RNA protein synthesize, mitochondria and chloroplast divide, where in S synthesis phase, G1 is the longest phase. Synthesis phase, DNA synthesize, chromosome duplicate and remain attached, and third, that is second growth phase, that is called as G2 phase, where the RNA and protein synthesize. Then we have seen that mitosis cell division, which include prophase, metaphase, anaphase and telophase, and last telophase is separating into two daughter cell with the help of karyokinesis. Karyo means nucleus, kinesis means division, and cytokinesis, cyto means cytoplasm, kinesis means division. By that, one parent cell is equally divided into two daughter cell. If the parent cell is haploid, the daughter cell will be haploid. This is called a mitosis cell division. Thank you.